And I just want to say it's so great to see you again, Bridgette. It's been a while.
Yes, it has.
And so Bridgette was one of the co-PIs on the AC squared grant, which will officially end now at the end of December, after 6 years, 5 years finally. And I just want to say that Del Mar was on the grant, Collin College was on the grant, Kentucky was on the grant, and they're not here, but Bridgette and her team were instrumental in creating the core course credit, and we really appreciate that. She was extremely dogged about that, and she organized all of us. Who knew that community colleges, when we partnered together, could actually get the state to turn around? They were actually going to say no to us, and Bridgette and the group met together in Austin and went to a meeting, and we changed that. So appreciate that, Bridgette. Well, anyway, Bridgette is going to be talking about incorporating biotechnology into biology programs, and they authored a lab manual. So take it away, Bridgette.
Okay, thank you.
And I hope your horses are doing okay and your dogs don't die. That's a whole nother story.
Whole nother story. Yeah, me too. My horses seem okay. I went riding this morning, so I weathered the cold so that I could be back for this. So thank you guys. We've had a lot of great talks, and this kind of has worked a lot into what I'm going to talk about today. Some of what Maggie and everybody else has been talking about. So traditional biology labs, they have, cookbook labs. They often lack real world relevance to students. And Vision and Change called for us to make changes in those. And really at the state of Texas, they've called for us to make changes to that, but we're still behind is what I would say to that. So how are we going to do this? And a lot of what we can do is befriending our biology faculty. Linnea mentioned that we incorporated some of this into our lab manual for our biology students. We're still piloting this. Of course, that has been put on hold because of COVID. But Carol Twitchell and myself and John Lawson began this project as we were doing some training with HHMI for C-phages. And and they're presenting all of the data about vision and change and the importance of vision and change and the importance of keeping students interested in the sciences by giving them something relevant and giving them real world experience. And at that point, we decided we couldn't live with ourselves anymore without trying to make some sort of change. So we began this project with the lab manual. And I would say in hindsight, it would be really important to befriend these people and get them on your side while you're working on doing this. I have lots more gray hair from all of that. But I started this by teaching an honors biology class. And that's one way to get your foot in the biology door if biology and biotech are not in the same department. Short lectures, helping the biology faculty incorporate those short lectures. One of the big things is I think that, a lot of the cookbook labs, they do use, they're testing for macromolecules, they're doing pH, they're doing a lot of these things that we think are important and we know that are important, but they don't have the why is that important? How do you use that in the real world? Why do you want to test for sugars, substrate, product, proteins, etc.? And a lot of the biology faculty may not be familiar with how those are being used in pharmaceuticals, in biomanufacturing. So those kinds of things are really important for us to be able to share with our biology faculty. I think right now during COVID, we have a novel opportunity with all of our Zoom meetings. And of course, here we are, and we're all getting kind of zoomed out. But I think traditionally, if you have to take a whole Friday afternoon or you have to travel to a different campus or to a whole different location, that that inhibits a lot of the professional development that can be done. And this provides us with an opportunity to share some aspects of what is, I was at this workshop for the last 2 days, and here are some things that might be relevant for our classrooms and in our labs that I would like to share with you. Maybe a 1-hour Zoom meeting for professional development. And I think a lot of our colleagues have really craved that kind of relevance. They just don't know where to get it and how to get it. We have been piloting this amylase cellulase lab for 5 semesters. Carol's nodding at me. Thank you, Carol. And it was really, really being opposed for a while. And then when the other faculty began using it and saw how excited their students were, it's really made some changes since we started, but we still have the amylase portion and going off of Ellen Doherty's amylase plasmid. But we have a product, and we test for the product. We test for we do both what Ellen does, which testing for substrate and testing for product. And we've developed some nice ways of doing that. And then later on, they'll do the same with cellulase. We actually, Carol and I think Rob and Daisy were at the same mini Bioman workshop with Jim Hewlett, in which we used the Biorad kit for biofuels. Yeah. And we got really excited about, you can, all these are cellulitic, and going through the woods and picking up different mushrooms and stuff that were cellulitic. And so we then started a project with using bacteria that have cellulase activity. And that's the second part of our lab manual. So they learn a lot of stuff in the beginning that they're gonna in the amylase part, and then they reuse that again at the end of the course, and they're repeating a lot of things. But the students have really got a lot out of it in that they see some relevance of why do I need to test for this? How is this going to be used in the real world? And I think, again, we can bring that information into why do I test for reducing sugars? What is a reducing sugar? Why do I test for proteins? And I think that it's more enjoyable for the students and the teachers alike. Carol and I have been chatting. Thank you guys for the last portion of the bio chain, the supply chain, and we were talking about how we could use that to recruit into biotechnology. In that, if we you have our students making some of the products for our biology labs, that we could use that as a recruiting tool, because our students' names would be on there, and the biology students would like, this is manufactured at Collin College Biotechnology program, and then they would see that, we have a biotechnology program. I don't know about y'all, but our biotechnology program is the best kept secret ever, not on purpose. So the other thing that I think that is really important is advocating for change at the state and local education level. Linnea brought up the fact that we advocated for making our first 2 biotechnology student courses academic as well as workforce. And we're able to, it was a long fight, and it was a longer fight to keep them as academic. But I think we have to emphasize the importance of biotech and how it affects our lives. And we need to work at the state and local levels to make sure that our education agencies are aware of the importance of these. I think we can also help to make them aware that some of these things are not really being hit on in traditional biology classes. So in Texas, we have what's called the 60 by 30. I always joke it's 30 by 60, but 60% of the workforce is supposed to have workforce skills in their degree, so that by 2030, and they should all be able to be employed after they have a 4-year degree. So you would think that that means you're going to change your traditional courses, but instead what they did was they went back and they looked at the learning outcomes of those courses and what's being taught, and they just called things workforce skills. So I think that we really need to be part of the advocating for changes in courses. We need to partner with universities to make sure that they are advocating for those as well.
Well, Bridgette, so you were part of that field of study up there, and they just shut down any kind of second year course as being part of the field of study for biology.
Well, and I think, you know, we have to be there to lay the groundwork, you know, and I did my part, I have to say, because I strongly advocated for biotech. And I, you know, tried to use their own arguments against them, which were we live in a molecular world. And I'm like, well, then maybe we should put some molecular biology in our curriculum. It didn't happen the first time, but maybe it's sowing a seed. And I think that having these university partners, Texas A&M, Corpus Christi, Texas A&M, like your partnership with Susan, those things are really important so that they can advocate on our behalf as well whenever the time comes. And the time's going to come again that we're going to have to make these arguments. But I think nationally, we need to make these arguments and work towards changes, that our biology courses aren't just history classes, that our freshman biology classes aren't just history classes, that they're actually, how does this affect the real world? And I think COVID is, again, we're cursing COVID, but I think it's also bringing science to the fore, and people learning that they need to know something about science for their own protection. And so I think that those are kind of important aspects. And
yeah, so they don't think that 5G cellular radiation causes the spread of the virus. That's a good thing.
Right. Exactly. So I do my share of cursing COVID, but I also, there are things in which I think that we can use it in to our benefit and to the benefit of our students. So a few ways of incorporating these into our courses and our biotech into biology courses. Again, you have to befriend some of your colleagues to get them to agree to this. And again, teaching an honors class or something special, a special class might be useful. Ellen Doherty, she has how to basics for biology classes. And again, we use amylase from her projects to do this. And Jim brought up yesterday the proteins, the cache, again, letting students see that there is a real world use for these types of things. Biorad kits, we use the Biorad Explorer, the DHFR kit, the BioENZYME, and even the GFP. And it depends on the amount of time that you have. In my Biotech 2 classes, I use the DHFR. I think Carol uses the biofuels for the 14 class, but you can go from either transformation all the way through to protein purification if you have less time. You know, the discussions yesterday were GFP kits. Sometimes the color leads students to believe that everything has a color to it, which is not exactly right, but it still gives them the idea of what, and GFPs and are fluorescent proteins have always been exciting to students. So, and making their plates that are, you know, have Christmas trees and stuff are always nice. This was a new one. When I went online and I did a search for biomanufacturing, this amino labs came up. I'm sorry Nick's not here today because I know that he was discussing last one of our meetings that we had that trying to, if you're not meeting in person, how do you get some labs to be sent home? These were kind of, they're within the realm of being affordable. Also, if you do the trialer simulations, there's some virtual bioengineer simulations, not too bad. And they have some good information. I have not used any of their resources, but they're probably worth looking at. It was created from somebody who, a PhD student who wanted to be involved in teaching, and this was her, after graduation, this is her means of doing that. Maggie brought this up earlier, the Bioscience Network. I've used these extensively, and we've incorporated some of these in our biology labs as well. Whenever we're doing the piloting, these these interactives are really good sources for students to be able to see what really happens, what's going on at the in the real world, and how they can apply what they're doing to some real world activities. This is a great resource if you haven't used it at all. And then this EDX, I haven't done any of these. Maggie brought up some really good sources this morning that I'm going to look into doing. Merry Christmas to me. Yes, that I want to do some more courses. But this particular website has lots of different courses. Many of them are free to be able to use. I don't know if anybody be interested to know if Jim DeClos or anybody has done any of these courses that are available.
What is EDX? Is just,
it's lots of different courses here. Oh, I can go there. I think. Let me see if I have to stop sharing. Can you see this or no? Or are you looking at my slide still?
I'm looking at your slide. I think you have to unshare and then share again.
Let me do that. It's really a pain, but you can find courses from all these different MIT, Harvard, Berkeley, University of Texas system. And I think I did a search for the biomanufacturing, and you can, the idea for me was that I could potentially use some of those in my some of those modules in my courses, and as part of teaching, especially as I'm looking at what changes I want to make for the quality assurance course, then what I can do there and incorporate for our students, because some things are kind of short little blurbs, and I think will be useful incorporating from a distance. Hopefully we don't have too much more distance learning, but we'll see. I do look forward to going back through the COVID talk again and figuring out. That was a lot in that talk, but it was a really good talk yesterday. So as our discussion, how are you all incorporating biomanufacturing in your courses? What else can we do? I like the supply chain idea.
Mm-hmm.
Discussion. Anybody have discussion? Let me read what's in the. I can't read what's in the chat while I'm doing.
No, I mean,
The HFR. Yes. Has anybody else used any of these in their courses?
I was going to say that you talked about the Delft course, so I completed that. That one is very much non-pharmaceutical biotech, and so you look at the stoichiometry of how much carbon comes in and how much CO2 goes out, and you do all of those calculations for the optimization of a process, because in non-pharmaceutical biotech, like we talked about yesterday, you need your margins to be right there because the products are relatively low value.
Okay. Interesting.
But it's a graduate level class, so it requires a lot.
Okay. Good to know. I may.
Jim, is that from Delft, the Netherlands?
Yes, from the Netherlands. There's another series of courses from, I can't pronounce it, but there's a technical university in the Netherlands, Vingen, and they're really into bioprocessing in the Netherlands, and it's part of the environmental ethic of producing products using biology rather than using petrochemicals, and so the Dutch are really into that. But the Delft one is interesting, of course, because they have the history of industrial microbiology that goes back to Leeuwenhoek. And so they do a lot of things of selecting organisms using selective media. And they invented this idea of microbiology selective media. And so they're still applying it here in an industrial process.
interesting.
Yeah, thanks, Jim.
So that'll be really what synthetic biology is going to be about.
Bridgette, Stacy. So in my biochemistry class, I do not use any kit, but I designed a LDH expression project. Students can actually isolate lactate dehydrogenase from a chicken breast. And then we test, it's very easy to test the enzyme activity. And then if I do have enough time, I let them start with the PCR, getting the, LDH gene cloned and try to see if we can, express it using some expression vector. So I pretty much just put it together, and they found it very interesting, very close to a lab version of biomanufacturing.
Very cool.
Thank
Hi,
you.
Marcy.
Hey, how are y'all today?
Good to see you. So Amy is heavily involved in algae at this
cool.
point, and manufacturing, and working with ATEC on manufacturing of algae products.
Nice. It is very exciting to see where everything is going now. It is just, it's amazing. Sorry, my computer keeps trying to tell me to do other things. I don't.
And Jim, you're involved with ATEC too. So we haven't even talked about the algae group with this group at all. Jim is involved with everything.
It's a really fun group. But our project is heterotrophic algae, the growth of algae heterotrophically. So what's interesting about that is you think of algae as being photosynthetic, but you can feed them as well. And so you grow algae in the dark, just like they're another organism. And by feeding them, your cell densities are greater by 200-fold. And they grow about twice as fast. So anyway, if you have a product that algae are especially suited for, then that's an interesting one. And so Lene, you want to make a plug for the algae microbiology course? So there's a algae microbiology online course that ATEC put out.
Right. And I mean, we started working with ATEC, what was it, Purnima, 3 years ago? 4 years ago.
4 years.
And so we happen to be next to the University of Texas, UTEX, which has the largest algae collection in the world. And our job was to create all the molecular biology modules for working with algae, even though that's actually much harder than it seems, since they're not easy to break and they're all very, very different. And so to me, it reminds me of an emerging technology was 20 years ago with bacteria. And there's no kits for this, trying to figure out how to break each one open and get the DNA out and barcode it. And Amy is now taking up the mantle for us in terms of working on this. And we have several people in lab working on this. And we have a little area cordoned off because, you know, especially after we listen to Joey, we do keep our algae separate from everything else to some degree.
And Danny Kaner has a great algae program in Lone Star.
Yeah.
So we went to visit him, and it's amazing what they've done. And then the other college that's heavily into this is Santa Fe Community College. So
Yeah.
they have a great program.
So are there modules from algae that you could maybe stick a biomanufacturing module in? Because many times our biology colleagues are not open to replacing their whole course, but they'll consider a module.
Well, that's exactly what happened.
Yeah, I'll let Amy talk about this because she's the one who's directing all this now. So go for it, Amy.
Thank you. So before I came on board, that's exactly what they did, is come up with modules to insert in each one of the classes that we teach.
Okay.
The barcoding, because of the contamination with E. coli, isn't quite as easy. And so there's some issues with that Shona at UT has been doing wonderful work on, and we're going to incorporate all her SOPs. But we are trying to bring more of algae into all the classes that we do in biotech, which we have control over. And I plan to have an entire process in biomanufacturing that we teach in the summer. The problem really is I would love to have an entire algae course in biology. And
Right.
that's where the difficulty lies. And I don't think that surprises anybody. I can't just wave my magic wand. But I think it's the future. And I think we have to make some of these courses more accessible in the biology department.
I agree. And I think that there again, I was kind of thinking about this. I was like, oh, we need to, spend some of our time being advocates. And then I was also then thinking, everyone in biotech I know has their plate like heaping full. I don't know what it is about biotech people, but our plates are over full of stuff to do. And that would be just one more. But,
it's really true that a college comes to me and says, I'd like to start a biotech program. And, then I immediately say, well, do you have any faculty that are willing to add a full-time job on top of their full-time job? And really, that's what it takes. And that's the amazing, and I don't think that's an exaggeration.
No.
I probably spend an equal amount of time with college duties as I do with my formal teaching classes, and that's kind of what it takes. And that's the amazing thing about this community is that each and every one of us are doing that. and it's incredible.
You know, one plug we could do for ATEC is maybe we should find the links to their courses. They have some amazing courses on algae, the large scale. What do you call them? You don't call them
Macroalgae.
macro, yeah, large scale.
It's a fancy
Kelp,
name.
kelp.
It's a fancy name for seaweed. Yeah.
Yeah. I mean,
Yeah.
okay, so
But
if you
it's
go,
it's a really fun group, and it always chooses Austin to
yes, they're
have a
MOOCs.
meeting, and they have meetings in San Diego, and the last one was supposed to be in New Orleans, and so it's a pretty fun group. So in Austin, if I can digress, in Austin, the project manager arranged Texas line dancing lessons for all of us. So on Saturday night, when they shut down the street I've forgotten what the street is where all the bars are.
6th Street.
6th Street. So they shut down 6th
Sixth
Street.
Street.
And so we're walking on 6th Street, and we start line dancing, and we turn around, and there's like 100 people. We initiated this flash mob, you know, and that's kind of what this group does. And it's a really fun group. But Linnea, remember 2013, we had, I think Sonia arranged us for all to go to Hawaii for a algae biofuels conference.
Yeah, that was really tough to go to. That was really hard to have to go to Hawaii. Yeah, because that's a big deal in terms of biofuels in Hawaii. It was a great trip.
Well, it was. So that's the problem, right? That I thought, oh, this is going to be a big deal because the Department of Defense is behind it. And so I didn't know anything about algae. So I signed up for the Algae Association of America online class, and San Diego State is big into it. I learned about algae, and then like 7 algae biofuels companies go out of business in the same month. And with the biofuels side, it's just, you can't compete with petrochemicals that have a $3 trillion and a 50-year head start. You can't compete with oil when it's as cheap as it is now. So,
but you can compete with algae products, because
with ultra high value products,
ultra high value products. So that's so. But it's a good way to teach biomanufacturing with an organism that doesn't have the same requirements. So you can teach biomanufacturing with algae.
Yeah, so Maggie has posted in the chat the link to the report that came out of the Hawaii meeting.
Yeah. And Maggie, we should probably, I don't know if Amy has shared everything that Shona has. Shona at UTEX has a lot of the SOPs for growing the different algae and doing molecular biology. And
we just talked about it on Wednesday, and Shona just gave the go ahead. And so there's 3 of us that are doing beta testing. So, you know, she still wants somebody to try them out. But she said we can put them up. So yeah, they're hot off the presses.
One thing Shona is doing that's really interesting is she has what she calls visual SOPs. So she's doing videos that are the SOPs. And cool. I've never heard of that, but I think it's an interesting idea.
Purnima did that with her, because first there was me, then there was Purnima, and then Purnima gave it to Amy. So Purnima, aren't you in some of the visual SOPs?
I was the cameraman and designer.
Okay.
You did visually seen. So, but yeah, we started doing these picture SOPs because there are some techniques that are hard to put just on description and actually get it done right. So having a visual would greatly make the procedure, learning about the procedure more valuable. So
I see one question from Jan, and I'm going to have Amy in my group answer, or anybody else. They are wondering, does anyone know if you can freeze algae stocks, or do they need to be continually propagated?
You can't freeze them.
Pardon?
You can freeze them. You freeze it, and yeah. So the ATEX Center for Algae, most of their stocks are frozen.
they also go quite a long time without being touched, which is why, we kind of have a 3-prong approach, and algae is really very easy to work with. I did give my students a little algae sample, and one guy did kill it by putting it in his car in the state of Texas in the summer. So I don't suggest that. But yeah, algae is really fun because it's just so easy to work with.
So Amy, whenever you get some of those, especially when they're applied to biomanufacturing, I think it would be really interesting to see how those could be, a module is created for, say, 1407, like the systems biology. I think that's one of the harder places to drop in a relevance, if you will, and convince our biology faculty that could go in there. But I think I've convinced, again, another upshot of COVID is that, we've had to go a lot of online labs. And one of my 1407, the systems biology colleagues, has done barcoding. I got him hooked on that a long time ago with his own project, but now he does fern research, and we're going to try to help him get that put into his systems biology class. But now that he knows that he can do online because he's had to, then he's going to stick some of his other stuff that he couldn't possibly do away with. And so he's going to stick in the systems biology. He uses the DNA subway. We've gone to a million years ago, we went to one of those workshops, and he does barcoding with his ferns.
So everybody should, are you going to get involved with metagenomics, Bridgette? So I don't know about how everybody else, I realize this is not biomanufacturing, but I can't stand the way they do systems biology where they just dissect everything. That gagged me with a spoon. I mean,
Sure,
seriously.
Totally. It's so grody.
I mean, seriously, it should be barcoding. And now we have phages, we have algae, we have metagenomics now out of Cold Spring Harbor. We can do water samples and soil, and we can do barcoding on plants, and they're still just dissecting everything. Who are these people?
And it's cheaper to do other things. Dissection costs a lot of money. They have to buy all the animals.
Yeah.
So if you teach, like many of us do, lower division majors biology, the cell molecular biology part is pretty interesting because you can bring in all of these techniques, and the students exit with real skills that they can apply. So when they transfer, then they can work in labs of the faculty. But the organismal biology part is caught in 1940 or something. Let's take out the prepared slide and look at the vascular cavity of the cross section of planaria, and you just go, oh my God. And what they should do is, just like you said, Linnea, they should do metagenomics. They should look at the ecological distribution of organisms and apply these tools. That part of biology has to be entirely reinvented. They have to throw out I want you to memorize the mouth parts of a crab.
There you go. The idea that we have to stop memorizing. My background is in physics. We did not memorize anything in the 4 years I went to school. And what I find difficult about biomanufacturing is that if the expectation is everything is worked out for you, you will not be prepared to do biomanufacturing because there will always be something that goes wrong. So I don't know how you get an entire, paradigm shift on education.
This is where I was saying we need to be parts of those advocates at higher education agencies, because it's hard, and they're pretty set in their ways about
Errol's cat,
what they're going to teach. But it's really like a history class more than it's a biology lab. And one of the things I did was I taught a couple of honor sections lab and I did DNA barcoding, and that was all I did with the systems biology lab. And the kids, of course, thought it was great, but my colleagues didn't really think it was great.
So Bruce must be gone. Yeah, I knew Bruce wasn't going to be able to stay for the whole thing because, you know, they have an IUS and metagenomics, and we're going to put in metagenomics in our biotech program in molecular biology. In our advanced molecular biology course, we're putting in metagenomics. So
we are, again, I hate to interrupt, but I have to.
I know we're off talking about other stuff. We have the student panel, which has already shown up to some degree.
I will stop recording at this point.